This video brings the second part of plant nutrition, factors affecting photosynthesis. Factors affecting photosynthesis are of two types, environmental factors and internal factors. Environmental factors are supply of raw materials like carbon dioxide and water, and outside factors like sunlight and temperature. Internal factors could be the quantity of chlorophyll. Testing a leaf for starch. Leaves produce glucose during photosynthesis, and some of the glucose is used by the plant and the excess is converted into starch. If starch is present in a leaf, that tells us that it has been photosynthesizing. Iodine solution can be used to test for the presence of starch. Testing a leaf for the presence of starch has to be done in few steps, and we have to be very careful about our safety during this test. As the diagram shows, testing a leaf for starch involves a few steps. Now let's see what are these steps in detail. First, we have to find a green leaf from a healthy plant. Boil some water in a beaker and drop the leaf into the boiling water and leave it for about 1 minute. This step is needed in order to break the cell membranes. The next step involves an alcohol, usually ethanol, which is highly flammable. Therefore, before we go to the second step, the Bunsen burner should be turned off. Put some alcohol into a boiling tube and place it in the hot water. Remove the leaf from the hot water using forceps and carefully place it in the tube of ethanol for a few minutes until the leaf loses its green color. Chlorophyll is not soluble in water, but it dissolves in alcohol. Therefore, all the chlorophyll in the leaf is now dissolved in ethanol. We can see the leaf turns usually white color, and the color of the ethanol also changes. Carefully remove the leaf from the ethanol as it is brittle now and dip it into hot water again to soften it. Then spread out the leaf on a white tile and add brown color iodine solution onto it. If iodine solution turns to blue-black color, it indicates the presence of starch. The diagram shows two leaves. The left side one has turned into blue-black color because starch is present. The right side leaf just has brown color of the iodine solution because starch is not present. Investigating the need for chlorophyll in photosynthesis. To show that chlorophyll is essential for photosynthesis to take place in a leaf, we can use a variegated leaf, which is a leaf with some green areas as well as white areas. Green areas contain chlorophyll, but white color areas does not contain chlorophyll. Once you select a variegated leaf, you can make a drawing of it marking the white and green areas separately. Then test the leaf for starch and compare your results with your drawing. Observation: only the green color areas of the leaf has turned blue-black. The white color areas remain brown, which is the color of iodine solution. So according to our observation, we can conclude that chlorophyll is needed for photosynthesis. The green color areas contain starch because photosynthesis has taken place there. White color areas does not contain starch because no photosynthesis has taken place there. Investigating the need for light in photosynthesis. We need to select a healthy potted plant because we need to keep the plant in a dark room for 48 hours. This will remove all the starch present in the plant, which is called destarching the plant. Then use a black color paper to cover a part of a leaf as you can see in the diagram. Once you do that, bring the plant outside and keep it in a place with plenty of sunlight for about 1 day. Then remove the leaf from the plant and also remove the black color paper and do the starch test for the same leaf. Observation: only the areas exposed to light turns blue-black color. The area which was covered with the black color paper remains brown. This concludes that sunlight is essential for photosynthesis. In the absence of sunlight, photosynthesis does not take place, thus no starch is found in the area which was covered with black paper. Investigating the need for carbon dioxide for photosynthesis. For this also we need to have a healthy potted plant which has been destarched. Set up the apparatus as shown in the diagram. Actually, setting up this apparatus is a little bit hard when you are going to do this practically, because we need to insert two leaves in two flasks with rubber bungs. Flask A contains potassium hydroxide solution which absorbs carbon dioxide. So, leaf A does not get carbon dioxide. Leaf B acts as a control. It is also inserted into a flask which contains distilled water. Rub petroleum jelly over the rubber bungs to prevent any air entering or leaving the flask. Otherwise, if the flasks are not airtight, our practical would not be successful. As potassium hydroxide absorbs carbon dioxide, the leaf in the flask with potassium hydroxide, which is leaf A, does not get carbon dioxide. Then keep the plant in sunlight for 1 day and test both leaves for starch. Observation: leaf A does not turn blue-black, but leaf B turns blue-black. Conclusion: leaf A does not contain starch as it did not get carbon dioxide for photosynthesis. Leaf B contains starch as it got carbon dioxide. Thus, it is clear that carbon dioxide is essential for photosynthesis. Investigating the effect of varying light intensity on the rate of photosynthesis. So in this activity, we are going to measure the rate of photosynthesis by changing the light intensity that the plant is getting. First thing, in order to measure the rate of photosynthesis, we are using an aquatic plant. So while the plant is in water, when it produces oxygen gas, we can see oxygen bubbles are coming out of the plant. Therefore, it is easy that we can measure the rate of photosynthesis simply by counting the number of bubbles per unit time, per 1 minute, 2 minutes, or maybe per 3 minutes. Or else we can collect the gas into a syringe, or we can use the water displacement method to collect oxygen gas onto a test tube so that we can measure the volume of gas produced by the plant per unit time. In order to change the light intensity received by the plant, we use a lamp to provide light for the photosynthesis. When the lamp is kept closer to the plant, the light intensity is high. When the lamp is moving away from the plant, the light intensity is getting lesser and lesser. Now let's see how we can do this activity. Set up the apparatus as in the diagram. We can use an aquatic plant like hydrilla. A length about 6 to 7 centimeters should be used, and it should be fully immersed in water where the cut end of the aquatic plant should float upwards. This is because we can see oxygen gas bubbles coming from the cut end very clearly. Now place the lamp 10 centimeters away from the beaker, and once the lamp is placed, wait for 5 minutes for the plant to adjust to the new light intensity. Then we can use a stopwatch and count the number of bubbles released from the cut end of the plant in 1 minute and record our results. We can repeat this 2 more times, which will make our results more reliable. Then move the lamp 10 centimeters further from the plant at a time and repeat steps 3, 4 and 5 for 4 more times. Once you are done, you can calculate the average number of bubbles per minute for each distance of the lamp. Results. This is a sample result table. We can see that more oxygen bubbles are produced when the lamp is closer to the beaker, and when the lamp is moving further away from the plant, the fewer and fewer bubbles are produced. So these results can be used to draw a line graph. When we draw the line graph, we should know what are the variables. Independent variable, which will be marked in the x-axis, is the distance between the plant and the lamp, which in other way indicates the light intensity. Dependent variable is the average number of oxygen bubbles given off per minute, which in another way indicates the rate of photosynthesis. Control variables are the type of plant and the size of the plant and the volume of water used each time. So when you use your results and draw your line graph, you get a graph like this. According to the graph, it's very clear the higher the light intensity, the greater the rate of photosynthesis. Limiting factors. A limiting factor is a factor that is in short supply which stops an activity happening at a faster rate. For example, the rate of photosynthesis is limited by the limiting factors such as light intensity, temperature, concentration of carbon dioxide, etc. At any point, the rate of photosynthesis can be increased by adding more light, heating towards optimum temperature, and providing more carbon dioxide. But increasing one of the limiting factors will increase the rate of photosynthesis only up to a certain level. This is because a second factor becomes a limiting factor and controls the rate of photosynthesis. Light intensity. The graph shows the rate of photosynthesis versus light intensity. So you can see as the light intensity increases, the rate of photosynthesis also increases, but only up to a certain extent. At low light intensity, the rate of photosynthesis is low as light provides the energy for the process of photosynthesis. Early in the morning and towards the evening, the light is limited. Even on a sunny day, light may be limited to plants which are shaded by trees. As light intensity increases, the rate of photosynthesis will increase until the plant is photosynthesizing as fast as it can. At this point, even if light becomes brighter, the plant cannot photosynthesize any faster as the other factors are limited. Carbon dioxide. The graph shows the rate of photosynthesis versus carbon dioxide concentration, which also follows the previous trend. Usual atmospheric level of carbon dioxide is about 0.03%. The more carbon dioxide a plant is given, the faster it can photosynthesize up to a point. But then a maximum is reached as one or more other factors become limited. This graph shows the rate of photosynthesis versus temperature, which is different from the previous two graphs. This is because some reactions in photosynthesis are catalyzed by enzymes. If it is too cold, the rate of photosynthesis will decrease as the enzymes can't work effectively. At low temperatures, molecules have little kinetic energy. Therefore, few enzyme substrate complexes are made. Equally, at very high temperatures, what happens? Plants cannot again photosynthesize properly because this will denature the enzymes. The high temperatures would denature the enzymes, so the rate of photosynthesis is again low. Stomata. Plants often close their stomata if the weather is very hot and sunny to prevent too much water being lost. In such situations, carbon dioxide cannot diffuse into the leaf, thus photosynthesis cannot take place. Therefore, on a really hot and bright day, photosynthesis may slow down. Knowledge of limiting factors is useful in farming as the limitations due to these factors can be minimized. This will assure higher rates of yield. Keeping plants in a greenhouse will assure healthy growth throughout the year under controlled environmental factors. Greenhouses trap heat so the plants can continue to have optimum temperature. Artificial lighting and supply of carbon dioxide can also be provided. So under these conditions, plants will grow healthily throughout the year and provide a higher yield throughout the year.